So what we are doing now, we are working on the genomic characterization of metastatic breast cancer. There are some papers that have suggested that the genomic landscape of metastatic breast cancer could be different to the one of early breast cancer. And as we all know, what is going to kill the patient is the metastatic cancer, not the primary tumor. So we are having several efforts. The first one is to describe the frequency, the distribution of the common recurrent genomic alteration in metastatic breast cancer, and to see if they have some if they are associated with specific clinical outcome like poor survival or better response to conventional treatment. So that is the first part about the conventional common recurrent genomic alteration. Then the second part is drug development, is the development of targeted therapies, but also immunotherapeutics and DNA damage drug that match to each of these genomic alterations. So this is the second part, is drug development. Then the third part is to understand the biological role, the functional validation of genomic alteration that have been acquired in metastatic cancer. For example, when a genomic alteration is not present in early breast cancer, but becomes recurrent in metastatic breast cancer, then we investigate in the lab if this genomic alteration could be involved in the metastatic process. And then the point number 4 is to work on genomic evolution and genome evolution between the primary and the metastasis. And for doing that, we look at what are the genomic alterations frequent in metastatic sample that we cannot find in the bulk of early breast cancer, and we try to understand what has been the evolution of the genome that led to the appearance of this genomic alteration. So we have characterized the genomic landscape of more than 200 metastatic breast cancer using whole exome sequencing, and there are several new findings. The first one is the fact that there are some recurrent genomic alterations that are specific to the metastatic sample as opposed to the primary. We found 11 genomic alterations, and some of them have been characterized and could be involved in the metastatic process. When a patient is presenting this genomic alteration, then the outcome is worse as compared to metastatic breast cancer who do not present this metastasis-specific genomic alteration. So this is one first finding. The second finding we had by running the genomic landscape of metastatic breast cancer is that a subset of estrogen receptor positive metastatic breast cancer present with very high mutational load and acquired genomic instability and high mutational load, probably because of high expression of ApoBec. And we found that around 10 to 15% of estrogen receptor positive breast cancer, when they are metastatic, have very high mutational load. And now there are some attempts to see whether this patient could be more sensitive to immunotherapeutics, and there are some attempts to understand what has made this cancer becoming so mutated. And as mentioned before, ApoBec is one of the hypotheses, but there are several other ones. Then, what we are investigating, and for which we have some interesting preliminary result, is whether these genomic alterations that are enriched in metastatic breast cancer, but present at low frequency in early breast cancer, could have some prognostic value when they are present in early breast cancer. And indeed, there are some of these genomic alterations, when they are present in early breast cancer, are associated with a worst outcome. The first one is to find new targets that could prevent the development of metastasis. Because if we understand what are the genomic alterations that are functional and enriched in metastatic breast cancer and led to the development of metastasis, that could generate some work about preventing metastasis. The second application is to better drive the drug development in metastatic breast cancer by defining what is the genomic landscape of metastatic breast cancer. And obviously, the genomic landscape of metastatic breast cancer is really different to the one of primary breast cancer. Okay. And the point number 3 is to better understand which patient with metastatic breast cancer is going to have a very poor outcome. And this is very important because if we can identify some genomic alterations associated with a very poor outcome, then the drugs that target this genomic alteration could receive some fast-track drug approval. So finding which genomic alteration are associated with worst outcome, very poor outcome, is a major challenge to drive drug development.